Another method of optically sectioning a sample is multi-photon or two-photon microscopy. This is a very advanced technology where we are taking lasers that can deliver temporally and spatially lower wavelength photons that can combine in exciting the fluorophore because they are coming at the same time and into the same spot. And so you get excitation only in the focal plane of the objective. And you are inherently sectioning the samples through the excitation. There is another advantage of multi-photon microscopy because you're using longer wavelength photons. They don't interact with the tissue as much as shorter wavelength photons. That means that the excitation reaches deeper into the tissue, and because you are intrinsically sectioning for just photons coming from the diffraction spot, you can pick up any photon that comes back, whether it scattered in the tissue or not. So multiphoton microscopy is used heavily in imaging in live tissue and in thicker tissue. Microscope automation has really helped also our ability to follow events in living samples. And in addition, with the development of the green fluorescent protein for marking proteins inside cells or having cells glow intrinsically, we are now able to follow events over time through time-lapse microscopy. And there are still challenges in doing this type of imaging, but the advantages of getting the temporal context of what is happening to molecules in cells is undeniable. Usually live cell imaging setups are very customized to particular applications, in the sense that whether you are imaging cells in a dish, or you're imaging a live animal, or you're imaging dynamics that are very slow, you need to sort of make sure that the setup is particularly equipped for that particular application. So these setups are not usually shared across multiple investigators who are doing different things. It's usually a specialty setup just for one particular way of doing the imaging. But we have found ways of doing imaging at very fast time courses. It is even possible to see calcium spiking in neurons using live cell imaging. Or we can follow cells over days in tissues and see how they develop in animals over time. For live cell imaging, the development of techniques with green fluorescent proteins has really been revolutionary. So think of green fluorescent protein as a tag that you can add to your protein of interest genetically, and you can make that protein glow in the dark, and you can now follow that particular protein over time.